Well, okay, and welcome all. We have a wonderful collection today of board members and donors from the ADDF. I'm Mark Reufner. I have the privilege and honor of serving as the CEO of the Alzheimer's Drug Discovery Foundation. We have a wonderful update on the current state of Alzheimer's research coming from you today. This is incredibly timely, as last week there was an international gathering under the title of the Clinical Trials Alzheimer's Disease Conference, better known to those in the industry as CTAD. Particular significance with the event this year. One, because there's been a lot in the news, but two, those of you who've been supporters of ADDF for years know that just last year, 88% of everything we funded because of your wonderful donations went into clinical trials and about 12% into preclinical. But that overwhelming majority of what we're funding is all about clinical trials. We were actually a gold sponsor of the event this year, which started last Tuesday afternoon and ran through Friday. It was in Boston. It was a hybrid event, about 300 to maybe 350 people in attendance, probably 700 to 800 online across the world. And we are very, very fortunate today because we have two phenomenal doctors who are there for all of it. Dr. Howard Fillit, our co-founder, along with the Lauder family and Chief Science Officer at the ADDF, and Dr. Jeffrey Cummings, who's a director of the Chambers-Grundy Center for Transformative Neuroscience. And he's a clinical trials expert and runs his own phase 2 study on a repurposed drug. I suspect you might hear a little bit about that today. I will tell you both Howard and Dr. Cummings were a bit of rock stars last week. Dr. Fillit ran 2 panels during the conference. And those of you who tuned in a little early, you might have heard me chit-chatting with Dr. Cummings, who served on at least 3 of the panels. I think it was more, but he tells me it was 3. What we're going to do today, I'm going to turn this over to Howard in just a minute, and he and Dr. Cummings are going to speak for the next half hour plus just about what went on at the conference, the key takeaways, and then we'll open it up for questions and answers. We'll have about 15 to 20 minutes to do that and then stop at the hour. When you have questions, just put them in the chat feature. So if you just click on the chat button and put your questions through, and we will do everything in our power to get your questions answered. And if for some reason we don't get to them, we will get you answers post the hour we're together today. So Howard, as I turn this over to you, I also had the privilege of being at the conference last week, and if I had to come away with 3 headlines, and you and Dr. Cummings should talk about anything you want, but the coming of age of blood tests in Alzheimer's seem to be front and center. There also seemed to be a lot about aducanumab and amyloid and tau. And at the same time, there was a competing amount about what I would call the biology of aging, and that kind of starting to take a center stage. So with that as a kickoff, Dr. Howard Fillit, I give it to you, and you and Dr. Cummings take it away for us here.
Thanks, Mark. This was, I believe, the 14th annual Clinical Trials in Alzheimer's Disease meeting. And I've been going to them. I think I've probably Jeff and I have both gone to every single one of them, or most of them. And I always felt one of the criticisms of the meeting was that there were never any results, not too much on the way of positive data. And so often it was not it's the leading meeting annually on clinical trials, but it wasn't the most exciting meeting, I would say. And this year was definitely different. It definitely had a different flavor. There was a lot of results being reported, a lot of trials that were being reported in the pipeline that would report out in the coming year or so. So it was really, in my way of thinking, to me, it was a landmark meeting, I think, in the field, and it really reflected the progress that we've made in the field. And as Mark mentioned, there was a lot about biomarkers and how much progress we've made on biomarkers, and particularly about the blood tests. And there were some innovative studies. I think where I'd like to start and then ask Jeff for his opinion is that one of the headlines at the meeting was about the anti-amyloid antibodies. And there was one sort of non-controversial panel about the controversy around aducanumab or Aduhelm, the Biogen drug. But I think, and we saw some more data from that trial indicating that the mechanism of action to remove amyloid from the brain also correlated with a tau signal in the spinal fluid and in the blood, which indicates that, again, along this pathway of anti-amyloid therapies, there really might be a saving in terms of neuronal brain cell death and destruction. But a really interesting part of the meeting was results on a similar antibody that's being developed by Eli Lilly. And that antibody actually has a different chemistry to it, even though they're both anti-amyloid antibodies, the Biogen drug and the Lilly drug. The Lilly drug actually has a different chemistry and seems to be more effective. And so Lilly reported some of their results at the meeting, which I thought were really quite interesting. And also Eisai, a Japanese pharmaceutical company, reported some of their results on their anti-amyloid antibody, which is also a bit different. And Roche reported on some of their data. And I think taken together, these are all beginning to support the idea that maybe anti-amyloid therapy really does work. It certainly works to remove the plaques, but maybe it really does work to help people in slowing the rate of decline. And with that, I think for the first discussion about the most advanced therapies and what was presented at the conference, maybe I'll ask Jeff what he thought about the general feeling about anti-amyloid therapies and the kind of data that were presented there.
Great. Thank you, Howard, and thank you, Mark. Thank you to ADF. It's really great to be here supporting an organization that I respect so much, and Howard's leadership has just been extraordinary over the period of time that ADDF has been growing into a really major influential organization in the Alzheimer's disease landscape. I had the same sense that you did, Howard. This was a meeting that turned a corner, right? We have been trying so hard for so long to develop drugs that would affect the underlying biology of Alzheimer's disease and have that effect of slowing the progression or delaying the progression. And none of them seem to do so until this meeting and this year. And I just couldn't agree more. We now have 4 antibodies, and they all look, they're all dramatically reducing amyloid in the brain. We can see that, there's no question about it. We can see it with these terrific scans. And I think the clinical data are also increasingly compelling. So aducanumab was kind of controversial because these trials were stopped early, but the data that was harvested eventually supported the clinical benefit. With donanemab, which is the Lilly compound that you mentioned, they used a sensitive scale, which is very important in very mildly affected patients. And again, they were with a statistically significant slowing of decline. In the lecanemab study, which is the Eisai drug, again, we used both the 8S-COG, which was a conventional instrument, and the ADCOMS, which is, again, a sensitive set of measures for use in early patients. And again, you see the slowing of decline. So I think this is very consistent and increasingly convincing and really exciting. So I just came away with a lot of enthusiasm from this meeting.
Yeah, and I agree. And then the next sort of drugs in the pipeline that seem to be mostly in phase 2 from big pharma, so they're really being developed, are these anti-tau antibodies. But we kind of got less positive data out of the anti-tau antibodies, I thought. We had this hypothesis that tau, which is a molecule that's in the connections between neurons, in the wiring of the neurons, and also at the synapses, that this was spreading through the brain. The biomarkers indicate that tau correlates well with cognition because it's a neuronal degenerative biomarker. And there was a lot of excitement, I think, theoretically, in the anti-tau antibodies, which were meant to reduce and remove this molecule tau, which is in the tangles, which is the other pathological hallmark of Alzheimer's next to the plaques. And so there was a reasonably good theory around that, but the data that were reported at the meeting didn't seem as positive on the kind of supporting the tau hypothesis. I was a little discouraged about the anti-tau reports, the anti-tau antibody reports. What did you think, Jeff?
Well, for one thing, let's tie that back to the amyloid question, because I thought it was just so interesting that both donanemab and aducanumab showed reductions in tau in concert with the reductions in A beta. And just as you say, the relationship between tau and clinical decline has always been stronger than the relationship between amyloid and clinical decline. So it looks increasingly likely that the reduction in tau is leading to the reduction in A beta is leading to a reduction in tau, and that actually may be very important in terms of the clinical benefit that we're seeing. But just as you say, when you try to target tau itself with an antibody, that has proven to be largely unsuccessful. There was, in the semerenumab study, there was one positive measure, but to me, and I think to most of the people, it wasn't quite a compelling story. And there have been several negative tau antibody studies before this. So I think we haven't turned the corner on tau yet. But one lesson from A beta is don't give up. Let's keep going. Let's find the answer.
Yeah, and the biomarkers, like you say, the anti-amyloid antibodies move the tau biomarkers. But what I think was really interesting and emerging is that there's more than one tau biomarker. So what we have is that tau is a normal molecule in the brain. It supports the axons and the synapses, and it's a marker of neuronal degeneration. And we can measure it with a tau scan, actually, just like we have a PET amyloid scan now. There's a tau PET scan on the market now that was just approved for use around the United States. So now we have two scans we could talk about, two brain scans that are already being used in clinical trials. And as we know from pathology, tau correlates very well with cognition. So it's going to be a better biomarker than probably amyloid. Amyloid just kind of gives you whether or not Alzheimer's is there, but doesn't correlate with cognition, whereas the tau scans, we know from pathology and from the scans, actually correlates with cognition. But then what we see in the spinal fluid and in the blood now is a molecule called phospho-tau, which is basically a modified form of tau. But there's different kinds. There's phospho-tau P181, phospho-tau P215, phospho-tau P231, and they all seem to have different properties. So we already have an amyloid blood test on the market. We're already seeing how that's being used in clinical trials, which is another thing we could start to talk about, how there were reports of how these blood biomarkers are starting to be used, like in the AHEAD trial that Risa Sperling reported on. But also these tau blood tests, how they're being differentiated into different types that might have different use or specificity. And I wondered what you thought about that, Jeff.
Yeah, well, thanks, Howard. Let me just emphasize the importance of blood-based biomarkers. Until now, it was either expensive scanning or it was spinal fluid, which of course means having a lumbar puncture to remove the fluid. And understandably, many patients don't want that. So it's been difficult to get as much of the biomarker present. Biomarker meaning something we can measure in the spinal fluid or in the blood that reflects what's going on in the brain. And it's just fantastic now that we're getting all these blood biomarkers. So as you say, we have them for amyloid. We have p-tau217, which is being developed by Lilly, which seems to change very early, essentially with the amyloid burden in the brain. p-tau181 also seems to be changing very early with amyloid in the brain. On the other hand, p-tau 231 appears to be changing later. And then the scan that you described, which is really a scan of the neurofibrillary tangle formed by tau, is even later, coming on just before symptoms begin to appear. So not only do we have multiple reporters, but we have an emerging sequence of the biology changes in tau that are occurring in the earlier stages of Alzheimer's disease. So it's hugely, hugely informative. And I was so impressed with the Biogen data that the p-tau 181 had very robust correlations with the clinical outcomes. So I think these are reflecting in an important way what's happening to cognition in patients with Alzheimer's disease. And after all, in the end, it's all about the patient. And these are stepping stones to helping our patients. That's what we want.
And it shows us how, we can really do modern clinical trials. I like to call them modern for lack of a better word, but, how far we've advanced in our ability to use biomarkers to really do much better clinical trials in patients earlier. Even patients that don't have symptoms, there's some trials going on.
Let me say one thing about that, Howard. I'm so excited about this, but what we can see is that the amyloid goes very low. And frankly, I think as a group, a research group, we don't really know what to do with that. If it's very low, do we need to continue treatment, for example? But what we can see is that the p-tau markers begin to rise in the blood during that time when the amyloid is still low. And that may well be the indication that we need to continue treatment in those patients. So I think that the guiding of the management of the antibodies is also going to be very informed by these blood tests.
Yeah, that's a great point, Jeff, that came out at the meeting. Is that it's kind of like cancer in a way, the way we think about cancer treatment. We treat patients. We hope to watch their biomarkers with cancer. It might be an X-ray of the lung, but it might be obviously these days much more sensitive biological markers. But we treat them, we see their biomarkers get better, and then we wait and see if the disease recurs. And that kind of model is actually what we're seeing emerging with Alzheimer's disease, which is really interesting because it might be possible that we see the amyloid disappear after 6 months of treatment. And then, as Lily was suggesting and some others, you could actually stop treatment. And we saw data that really going out almost 2 years, there's almost no or very little increase in the recurrence of the amyloid, which means the disease can take a long time to come back and cause symptoms or, to create havoc in the brain, which really changes the clinical model, because I think the thinking that you had to go on infusions for the rest of your life was really quite something difficult to imagine, especially since we haven't talked about the side effects of these drugs, which is actually pretty serious, I suppose. And we saw some data on that. Jeff, I wondered if we could maybe move on to some of the other things that we saw. One thing that I was excited about was the semaglutide trial. And we haven't seen any data per se from that trial, but ADDF sponsored a clinical trial of a similar drug called liraglutide, which was on the market before semaglutide. The liraglutide drug is an injectable, and what it does is it basically improves metabolism and insulin resistance in people with diabetes. But of course, in the aging brain, we see insulin resistance, and we thought the theory from both epidemiological data and animal model data was that liraglutide could actually improve symptoms. So we sponsored a clinical trial at the Imperial College of London with Paul Edison, and we saw results on FTG glucose monitoring, looking at better metabolism from the drug and so on, which inspired this large Novo Nordisk's trial of semaglutide. And I'm pretty excited about that because, again, you have Byetta, which is one form of a GLP-1 agonist, which is the class of drugs we're talking about. You have dulaglutide, you have semaglutide, and you have liraglutide. And all of these drugs, both in epidemiological studies and in clinical studies, seem to show improvement. So there's a body of evidence that this kind of improving metabolism by improving insulin resistance could really work to slow down the course of the disease. And I was pretty excited to see the semaglutide trial because semaglutide is the only one of those drugs that's orally available. But I don't recall seeing any preliminary results or biomarker results from that. I guess I'd ask you what you thought about the data presented on semaglutide, if there, or the whole proposition of semaglutide as a potential drug for Alzheimer's. And, it's not just in diabetics. This is a drug that could also be used in non-diabetics to slow the disease down.
Yeah, I agree completely, Howard. I mean, it's just so exciting to see this new class of drugs coming in. There were no data presented. They presented a lot of the background of the trial. And one of the things that I found particularly interesting is that there are now 4 registries where people have been followed over time, mostly diabetics being followed for cardiovascular disease. And all 4 of them show a reduction in the emergence of Alzheimer's disease. And one of them, the reduction was on the order of 50%. It was very robust. So I share your enthusiasm for this. I think there's animal data that supports the GLP-1 agonist. There's epidemiologic data that supports their use. We know that there's insulin resistance in the brain. And the main effect of semaglutide may be on inflammation in the brain because diabetics have a lot of inflammation. And when you give them semaglutide, you reduce the measures of inflammation like C-reactive protein. So we know that's happening, and that may well be a key to what they're doing in the brain as well.
Yeah, that's a great point. I wasn't thinking about that, but when the presentation, they actually postulated neuroprotection and an inflammatory effect as part of the way the drug worked. And it's a good example. I don't know that people really thought about it, but it's a good example of repurposing, because this is a diabetes drug that's being repurposed for Alzheimer's disease. And maybe that's a good segue to talk a little bit about our panel, which I thought was great. Of course, we were both in it, so we're going to think it was great. But we had you presented on the study we supported on rasagiline, and Anna Pereira talked about her studies of riluzole, and Giacomo Koch flew in from Rome to talk about the studies we supported with his drug, which was repurposing a Parkinson's drug called rotigotine. And the whole purpose of the panel was not just to report the results of these drugs, an ALS drug and 2 Parkinson drugs, but also to talk about commercialization, because the theme of the panel was very often in repurposing, you see an academic study reporting the results of a small clinical trial, repurposing a drug. It goes into the medical literature, and then it kind of dies there, and nobody prescribes it offline. There's a lot of off-label. There's a lot of challenges in prescribing off-label these days. Insurers don't like to pay for it. And I thought we had a really good discussion about not just the results of these 3 ADDF-supported repurposing studies, but also where do we go next? And you talked about your pathway, and maybe you could comment a little bit on the panel, because I think we got a lot of kudos on that panel particularly.
Yeah, I think it's really important. Repurposed drugs, just to be clear, are drugs that are approved for one disease, maybe Parkinson's disease or ALS, in the drugs we're talking about. And then they have promise for use in Alzheimer's disease. So they are repurposed for use in Alzheimer's disease. But of course, they're not approved for use in Alzheimer's disease. So one could use them off-label, but you can't market them, and you don't have as much information. The importance of repurposing is that much of the original work is already done. The animal work is all done. The dose finding is all done. Phase 1 safety is all determined. So you essentially begin the development program at phase 2, cutting off at least half of the length of the development program. That is so important. We need drugs fast. So if we can cut the time down by 50%, that's great. But it is true that very few of those drugs have made it into phase 3. And Part of that is the intellectual property problem. That is, maybe they're already generic, and so pharmaceutical companies have shown less interest in them. But the panel was so instructive because you could see that by adjusting the dose to a different dose, you might be able to get intellectual property. By adjusting the formulation, you could get intellectual property. You might be able to change the molecule slightly, and that would give you intellectual property as a new molecule. And certainly there's tremendous learning in those trials. You learn about biomarkers, clinical outcomes, new designs, all the things that we need to know to advance the field in general. But specifically, I think we can advance those repurposed compounds if we're clever about how to manage the intellectual property.
Yeah, and there was good discussion around that. And I forgot to mention Michaela Gallagher, who we've been supporting and investing in her company, Aging Bio. And she was an example of how she got intellectual property by changing the dose to like 1/10 of the normally used dose in a reformulated type of pill for her drug, which is for mild cognitive impairment. And the clinical trial, it's essentially a phase 2b, phase 3 study. If it works, she might be able to bring that to market. And she's been at this a long time and developed a use patent and a very strong intellectual property. But she hasn't been able to get a lot of investors and has really kind of run the company on NIH grants. And NIH, of course, has a lot of money these days. So she's gotten something like upwards of $100 million to run the study. With Giacomo, we're trying to create new intellectual property by creating a new combination of rotigotine, which is his drug, along with one of the old existing drugs, rivastigmine. They're both patches. And so we eventually can envision a day where the new intellectual property would be one patch that had two drugs that had different symptomatic effects for patients and that were additive. And of course, Anna Pereira has a really exciting drug, in my view, Riluzole, which could replace one of the existing drugs on the market, memantine, which is also one of the drugs that's on the market today for Alzheimer's that comes from repurposing, because it was originally a drug for another indication. And somebody came up with the idea that it was an antiviral being distributed in Europe. It was sold in Europe in the 1960s as an antiviral. And someone came up with the idea that you could use memantine for Alzheimer's disease. And there was a big intellectual property fight. The generic companies got involved. But today, memantine, most patients in the United States are on two drugs. One is a drug like Aricept or donepezil, a cholinesterase inhibitor, and memantine. And memantine is a repurposed drug on the market today for Alzheimer's disease. So, we're very excited about our repurposing clinical trial.
I think the other thing that bears mentioning is that those drugs are less expensive. And that's why NIH can fund them reasonably. ADDF can have a major role in them, and that's important because if we can figure out how to get around the tremendous expense of drug development, we can have more drugs in development and eventually more drugs on market.
But just to put that in perspective, because we did do an analysis of what it might cost to bring in a repurposed drug to market. Excuse me. We're supporting phase 2 trials, maybe $3, $4, $5 million, and it gives us enough shots on goal that if they're interesting data, hopefully someone, NIH or investors, can come along and fund the next study. Because while the repurposing trials we're funding are in the phase 2, when we get to the phase 3, now we're talking 50 or 75 or $100 million. The overall cost of developing the drug might be 1.2 billion when you start from scratch, right? So as you said, Jeff, you can leapfrog that, and we can reduce the cost, but we're still talking about a lot of money, which would require, I think, investors or maybe some NIH, because again, they have 3.5 billion, but it's still not inconsequential, the amount of money and effort that's required to actually get that new indication and bring these repurposed drugs to commercialization and market and patient access. Another thing that I think came up, Jeff, was I don't know if you want to talk about Green Valley, which is one of the first new drugs approved in China and being developed in the United States. And I know you've been very involved in the Green Valley story. Maybe you could tell us a little bit about the Green Valley drug.
Absolutely. And I see that there's a chat question about senolytics.
Yeah.
And that really overlaps with the Green Valley story. So the Green Valley is a company that's developing GV, Green Valley 971, and it acts primarily on the microbiome to reduce inflammation in the gut. And we've already been talking in terms of semaglutide that systemic inflammation apparently drives brain inflammation. And if you can reduce the systemic inflammation, you can reduce the brain inflammation because some of those inflammatory cells cross the blood-brain barrier and many of the inflammatory chemicals cross the blood-brain barrier. So the idea of GV971 is to reduce the inflammation, which is present in the gut of many Alzheimer's disease patients, to reduce systemic inflammation, which has been measured and shown, and that will reduce neuroinflammation. So we're hopeful about this. As you say, there was a successful phase 3 trial in China. It was approved in China and is on the market in China. And now it's being studied in a global phase 3 program that is primarily in North America and Western Europe and China. So a really exciting program with, I think, a very interesting biology. And then that takes us to the senolytics, because senolytics are a class of compounds that interfere with the senescence of cells. And therefore have effects not only on the brain, but on many cells in the body. So in the senolytic trials, many of them include cognition as an outcome, but not the only outcome. So there might be cardiovascular benefits, there might be arthritis benefits, as well as benefits on cognition. So there are a few of these senolytics that are specifically looking at changes in senescent cells in the brain. And I think they're very interesting, and they might also have systemic effects as well. And to tie this back, Howard, to our earlier discussion, it looks like tau in the cell often leads to cellular senescence. So senescence may be a downstream effect of tau, and maybe we can interfere in that downstream process with senolytic compounds. So I see that as a really interesting, novel, and important theme that's evolving in Alzheimer's disease therapeutic research.
Yeah, and Miranda Orr from Wake Forest, who we funded to do a senolytics trial, gave a great talk, I thought, on her clinical trial, where she's repurposing two compounds, dasatinib, which is a cancer drug, and quercetin, which is a senolytic drug. The thing about senescent cells is when cells get old, they refuse to die. And when they refuse to die, they become very inflammatory, and they chronically release inflammatory factors that are killing cells in the brain. So it's interesting that senolytics are being used and being investigated for cancer treatment to help these cells die. But you want to kill cancer cells in the brain. The question is, like, do you want those cells to die? And you probably do. You want to get rid of them. But then, does that kind of mess up circuitry? I think was one of the concerns. It's kind of a theoretical concern, but we're really interested in the senolytics, and it really extends directly to our strategy, which is promoting the biology of aging. I know we have some questions here, so I've been instructed to start addressing some of the questions, Jeff, that we see. But I just want to encourage everyone that if you go into the chat, you can ask questions, and Jeff and I will try to answer them. One of the questions is, Jeff, about if we remove tau and amyloid, could that be a possible pathway to treatment? My personal opinion is that the way drug development works in combination therapy, which is kind of the question and where we're going, we think with Alzheimer's treatment, it's going to be combination therapy based on a person's biomarker profile. And so we have amyloid and tau and all these other biomarkers. We'll be able to identify people with those kind of abnormalities and then create a combination therapy. So this question is about combination therapy, but according to FDA regulations or guidance, to develop a drug that's a combination therapy drug, you have to have 4 arms. the placebo, one drug alone, the other drug alone, and the drug in combination. So you have to show that each drug works alone and then show that the combination is better. So if we're seeing amyloid effects, that there's some consensus maybe that we are seeing a marginal effect that, or maybe something more than marginal depending on the antibody. That's a good thing. Amyloid will someday, I believe, go into combination therapy with other drugs, but will it be tau? Well, if we don't see tau effects of tau drugs, tau monoclonal antibodies, then it's unlikely that there'll be a combination of amyloid and tau drugs, because again, according to FDA guidance, we need to see the tau drugs in by themselves having an effect. Jeff, I wondered what your thoughts were about this issue.
I think we're definitely moving towards combination therapy. That's been the successful story for tuberculosis, HIV, many cancers. So I think we're definitely going towards combination therapy. And there are many combinations that we could envision. And I know ADDF had actually a meeting on combination therapy. We've just been talking about inflammation. So it may be that a combination of an anti-amyloid plus an anti-inflammatory would be a very useful combination. But as you say, the FDA requires that the benefit of each drug be shown independently before they're used in combination or at the same time in a 2x2 trial of the type that you're describing. So these are complex developmental pathways to try to get two drugs to work. And it's also true that companies rarely have two drugs that have complementary biology at the same stage of development. So then that requires a collaboration, which has sometimes been difficult.
One of the questions is about some of the other things that were excited in the pipeline. And I thought there were two kind of interesting trials that were reported that got a lot of press and kind of really out of the ordinary. I think the Cortex-Zyme study and the Cognito study, which both, the Cortex-Zyme study got failed in its primary outcome. And so the Cortex company, which is publicly traded, and we looked at it and declined an investment. It's based on the idea that if you have periodontitis, if you have infection of your gums, that's a risk factor for Alzheimer's disease. And then it was discovered that the bacteria that causes periodontitis releases a factor called gingipain, which apparently gets into the brain, and the company had some good evidence that it does. So they created, and that this gingipain is neurotoxic. And so the theory was that if you had infection of the gums, which is very common in older people, not so much in younger people who actually are developing Alzheimer's preclinically. So that's one question I think we have. But that this drug that they developed that neutralized the gingipain in the brain could be therapeutically effective. And what they showed in their report, which was obviously in a press release as well, is they studied the whole population of Alzheimer people. And this is an interesting clinical trial model, actually. It's called enrichment using biomarkers. So they studied the entire population and they didn't see an effect. But they identified people by saliva who had positive studies for the bacteria in their mouth, indicating that this P. gingivalis, which is the bacteria that causes the periodontitis, and it releases the gingipain into the systemic circulation and into the brain, that the people who had evidence of P. gingivalis and therefore would have gingipain in their brain, they responded pretty well to their drug. And at the bottom line, at the very end, they also saw that their drug improved periodontitis. So if nothing else, you get a drug for your periodontal disease and your gum disease. But I thought that was pretty interesting, and the market seemed to really slam the company. But I thought there was a little signal there for, if you could test people for this gingipain or for the P. gingivalis bacteria, you could treat them to remove that and they might get cognitively better.
Yeah, I think it was a really interesting observation, Howard. And as you say, the main measures were negative. The trial did not meet its primary outcome. But just as you say, those that had evidence of the P. gingivalis seemed to respond to treatment. I'd want to know two things. One is, Was there anything else special about the group that had P. gingivalis that could account for that? So I think we need more data on that. And the other is that I'm still puzzling over the time course. That is, the infection that would drive Alzheimer's disease had to have occurred about a decade ago.
Exactly.
In terms of our understanding. So how confident can we be that finding P. gingivalis in the mouth now reflects what was going on a decade ago? And maybe it's very tight, that's a possibility. But I think there's still some learnings to do with that. But I think they're doing essentially the right thing, which is, okay, you've got a biomarker defined population. Let's see whether they respond. I would see that a little bit more as a phase 2 trial than as a phase 3 trial, but these numbers are somewhat arbitrary.
I see it's a good illustration of how comorbidities can worsen cognition, let's say in old age, and be a contributing factor that's treatable, as opposed to, as you said, a cause of the disease, because we know the disease starts, let's say, when you're 50, and most people at 50 don't have periodontitis, so it's hard to speculate that this is the cause of Alzheimer's, because we know the amyloid's accumulating, which leads us to another question that relates to really prevention. So with these biomarkers now, with the brain scans, and even more importantly, the blood tests, we clearly know that Alzheimer's starts maybe 20, 30 years before people become symptomatic. Although the earliest symptoms, maybe with really sensitive cognitive measures, we could pick up earlier than we are now. But there are clinical trials that are trying to remove the amyloid in people that don't have symptoms to see if by removing amyloid in people that don't have symptoms, you prevent those people from progressing to have symptoms. And this is really an amazing paradigm for prevention, which I think could really be very exciting going forward, although hard to prove. These have to be, by their very nature, very large trials. And I think the idea of using infusions for prevention is not a great clinical model. I think it would be much better if we had a little white pill. But I wondered what your take was in the reports at the meeting on this kind of treatment of patients without symptoms.
Well, wouldn't it be great if we could prevent people from ever having Alzheimer's disease, or at least the symptoms of Alzheimer's disease? What we're all looking for is a healthy, dignified aging, right? And that comes close to it, is if we can prevent that deterioration that occurs with Alzheimer's disease. And as you say, there's the solanezumab trial is just about ending. There are some other prevention trials, like in autosomal dominant families, that we're going to see reading out before too long. And then there's the new trial that ASI is collaborating with in a public-private partnership to introduce their antibody very, very early. We're going to have data on this. And I think that's another place where we're going to see tau be very important, the tau blood biomarkers, Howard, because people can have amyloid in the brain for so long that if that is your only measure, then they might have no change during the course of the trial. But if they have amyloid and rising tau levels, that's likely an indication that that patient is going to become symptomatic within the framework, a reasonable framework of a clinical trial. So I think we're going to be smarter and smarter about choosing patients in which we can see the effect of the drug, because they would change if they weren't getting treatment. And I just think it's so important. And prevention is always better than treatment, right? We've known that from other diseases as well. We don't want to wait for symptoms to occur if we can possibly avoid it. And
the ADDF has been investing in these blood biomarkers, as you know, and we did invest in C2N to bring, the blood test is on the market now. I've ordered it on several patients and I've seen the results, and I don't have to order the brain scan anymore because the correlation between the blood test and the brain scan is so high. So the blood test for beta amyloid, which is an indication of the presence of Alzheimer's disease in people with very minor memory complaints, is on the market in 49 states. It's not on the market in New York because we have the most restrictive qualifications, but a lot of my patients are outside New York or can go outside New York to get the test. And the tau test I think the tau test will be on the market in a year or two, and we're invested in that through the Diagnostics Accelerator, which is this $50 million effort that we have with Bill Gates and Leonard Lauder and David Dolby and Jeff Bezos and others. And we're just about excited about being able to announce the 2.0 version of that. Part of that effort was something else that was discussed at the meeting, which is digital biomarkers. And when we talk about early detection, so that we can pick up people in even the preclinical stage, using things like being able to do cognitive testing on your iPhone and picking up changes in speech and language. We have a big effort at the Diagnostics Accelerator in detecting very early abnormalities in speech and language, vocabulary, grammar, tone, the intensity of speech and language, has really early biomarkers. For those people on the call, and myself included, with my own father, I remember just kind of suspecting he was talking a little bit funny. Imagine that we'll be able to quantify that in a very rigorous way. And we're setting up a database now that will have thousands and thousands of people in it that researchers all around the world will be able to use to advance this field of digital biomarkers. And we're using digital biomarkers for so many other things. I thought there wasn't a lot, but for the first time, there were sessions on digital biomarkers as not only early detection and as non-invasive, very sensitive markers of the very earliest stages of clinical Alzheimer's disease, but also for their use in clinical trials and being able to do cognitive testing online, for example, and greatly, again, to the degree that the blood tests are going to reduce the cost and improve the efficiency of clinical trials. These digital biomarkers, I think, will do the same for clinical trials in terms of early detection, enrollment, and monitoring of the effectiveness of treatment. What's your view on the value of digital biomarkers in clinical practice and in clinical trials?
Yeah, I think it's another just great theme of innovation that's going on right now in the Alzheimer's disease world. We're seeing so much. One of the companies I'm working with, and I don't think they presented, is able to measure the strength and accuracy of keyboard strokes by people who are typing, and also the strength, the correctness and accuracy of your phone, use of your phone. And of course, you make more errors. You go slower as you have progressive cognitive impairment. So that kind of passive monitoring, which of course is done with the person's permission, there's nothing without informed consent. But increasingly, we're able to use these passive biomarkers, digital biomarkers, to detect early changes. And that's the population for which the new drugs are approved, are patients with very early disease. And we know that they go undetected for long periods of time, and they may go undetected until they're out of the treatment window. So we really need digital biomarkers and other means of early detection in order to take advantage of the therapeutic progress that's being made.
You know, Jeff, one of the questions that's in the Q&A is we didn't really get in much to the discussion of side effects of these monoclonal antibodies. They're being infused. And I know you and some other colleagues wrote some appropriate use guidelines for how to use these drugs in clinical practice, like aducanumab, for example. I wondered if you could comment on the prevalence of side effects like the brain hemorrhages and the edema, which affect up to 40% of people who are ApoE4 positive, and how we're going to manage those side effects in clinical practice.
Now, really an important consideration, Howard, because the last thing we want to do is to increase the burden of disease that the patient has through side effects. Now, most of these ARIA events are without any symptoms whatsoever. So they are detected on routine monitoring with MRI, which we do now because we're worried about these events. But only about a third of them have any symptoms associated with them. And the symptoms would be like a headache or confusion or some visual disturbances. Those are the things that are commonly reported by patients who develop ARIA. but a very few of them have severe symptoms like convulsions. And there has been one death actually since the approval of aducanumab. So we need to take this seriously. The management is pretty clearly defined. And as you say, we wrote appropriate use recommendations that really emphasize the safe use of aducanumab. We're going to have similar monitoring for all of the monoclonals, although they probably have different rates of ARIA. I think we don't have quite enough data to conclude that yet. And the other question that we had just a really great but unresolved discussion about is, just as you said, the people who have the apolipoprotein E4 gene are more likely to have ARIA than people who do not. And that's a gene that about 60 or 65% of patients with Alzheimer's disease have. So it's common. And it seems particularly risky for the patients who have 2 copies of that gene, and they have an even higher rate of ARIA. So I think one of the things that the scientific community is not settled on yet is, should we require the gene testing before we give the drug so that people can make a more informed decision? Now, the argument against that is that, well, we would monitor everybody with the same rigor. So we wouldn't actually change our monitoring behavior, but we might have different sensitivity to the headache that a patient reports or other symptoms that may be very difficult to ascribe to the ARIA. But if we know that they're an E4 carrier, maybe we would go ahead and get the MRI to make sure that we're not missing ARIA. I also think a really interesting area of drug development would be a drug that reduces ARIA. And we do have drugs that affect the integrity of the blood-brain barrier, which seems to be interrupted and to be a part of the ARIA physiology. So I'm thinking that we might even see the emergence of small molecules, which could be very useful in reducing the rate of ARIA.
The monitoring that you're talking about to do serial MRIs over the course of the year raises the price of the drug effectively from about $56,000 a year to closer to $100,000 a year. And we've already seen recently in the newspapers that the Medicare pharmacy budget has already been doubled. There's a huge increase in, I don't know if double, that's the right word, actually, I think it, but I read recently that there's a huge increase in the Medicare premiums for the Part D plans, for the drug plans, in anticipation by Medicare of the use of these drugs. And I think the cost issue is going to be a big barrier to utilization, because as Part B drugs, there is going to be a 20% copay, at least on the $56,000 a year. So even if people have, it's confusing and it's complicated, but I think the cost of the drug is really complicated by not just the cost of the drug, but the cost of monitoring the drug. I guess what I want to close with is something that you and I participated in, which was the value of exploratory phase 2A studies. We had a great panel, myself, you, doctor from Merck, and Suzanne Hendricks, who is a statistician and a consultant to a lot of companies on how to design these trials. And basically, we kind of talked about the ADDF's core strength and what we do in terms of funding exploratory trials. We kind of alluded to it a little earlier today. These phase 2A trials that cost maybe $3, $4, $5 million that look for signals and try to learn about the drugs, but they're not necessarily useful for getting regulatory approval. They're what the phase 2A trials or phase 2 trials that are useful for getting regulatory approval are called regulatory enabling types of trials that are very, very stringently monitored and the data is collected. We're talking more about the kind of trials that are conducted in academia and small biotechs, just to see for the first time if there's a signal, if the drug is working. And we had one report of a phase 2 exploratory trial from one of our companies that we're very excited about, Pharmatropics, where there was a lot of biomarker data, but there wasn't any clinical outcomes, and the very difficult question about go, no-go decisions. And I thought we had a great discussion. I know there was a lot of audience interest in response to that trial, that panel. And I'll let you comment on what you thought was all the panelists, including myself, obviously thought that exploratory trials are important to create innovation in drug development. But we need to be careful about how we use them. Maybe you could comment on that, and then we'll turn it back to Mark to close the session.
Absolutely. Several people came up and said, Jeff, that was the best panel of the CTAD. So I know that
I
people
had the same experience.
Of course, I'll just point out that it built on a great meeting that ADDF sponsored a few years ago or 2 years ago. And then this year, there was a publication that came out of that workshop in neurology, very prestigious journal. And I know a lot of people who have used that article to help them with decision making. So ADDF did a real service to the field with that. And I think we've learned a lot about exploratory trials. We want to do them rigorously, and we have to train academics to do them rigorously. But we also have to be more open to different kinds of outcomes rather than just pre-specifying 2 outcomes that make the difference. We're going to use biomarkers much more as the outcome and the report for those trials. There's a different kind of statistics that goes into them. So I think learning how to do those trials well is really important. And I think that panel went a long way toward defining how to do good exploratory trials.
Well, thank you, Jeff. And I want to thank everyone for listening. And I wish we had more time, but I'll turn it over to Mark.
Well, Howard, I may, first of all, excellent. Doctors Cummings and Fillit, just excellent. As a lot of the postings on our Q&A were shared with you all. I have one just kind of last thing to leave us on, and it dawns on me that Dr. Cummings, you do this once a year, and the ADDF does it once a year, and really scans all the Alzheimer's clinical trials that are out there. There's about 118 that are out there now. And Howard, ADDF's clinical trials report came out at the very beginning of this, of last week of the conference. And I was struck by, of the 118 trials that are out there, something like more than 3 out of 4 are not so much about the plaques and tangles and the amyloid and the tau, as much about this kind of concept of biology of aging. And I just, more than anything else, Howard or Dr. Cummings, just your sense of the whole field right now, what's going on in the whole field, and we can leave folks on that.
Well, I think the field's really matured because, in the old days, and I'm talking about even 3, 4, 5, 6 years ago, it was kind of like amyloid all or nothing, and tau all or nothing, and it was it amyloid, was it tau, blah blah, or was amyloid going to work or wasn't it? And we've been at this for a long time, several decades, including 2 decades at ADDF, to look at other mechanisms, other pathways, the biology of aging, inflammation, neuroprotection, autophagy, cellular death, and it's kind of gratifying, I have to say, to see this much broader perspective taking hold now. And I think, there really is an acceptance in the field that even if the anti-amyloid antibody and anti-amyloid approach works, it's not going to be the cure. And it may even just be a marginal or slightly better than marginal effect. And we're going to need combination therapy. So I think this meeting was really one of the first meetings where you saw a pretty good representation of other approaches where they're actually in phase 2, and we're actually going to see data. And I think we're going to see a lot of positive data from these other approaches. So again, it's a maturing of the field. It was a landmark meeting. And I think the world has changed in Alzheimer's therapeutics.
Yes. Dr. Cummings, last word.
Yes, I would just add to that this increasing repertoire of targets reflects the growth of the science in just such a great way. For example, we've learned so much in just the last few years about the genetics of inflammation and how that influences the occurrence of Alzheimer's disease. So the growth of that basic science information is allowing us more insight into the disease, and that's what allows us to develop new drugs.
And the biomarkers, if I might add, are really allowing us to do the clinical trials to ask these questions. So inflammation biomarkers, and all the rest, we have a whole panoply of new biomarkers beyond amyloid and tau that are going to enable us to do clinical trials to test these new hypotheses.
And on that, my good doctors, first of all, we have to thank everyone who tuned in today are our donors, because you literally are making that happen. Number one, so thank you to all who tuned in today. And really, Doctors Cummings and Fillit, you were excellent today. Thank you so, so much. And we look forward to hearing about next ye's conference. Take care.
We're good. All right. Bye.
Everyone.